Sometimes it costs like $500,000. Electrically stimulating the microchip. And real reason behind is they're losing dopaminergic neurons in the substantia nigra, where the midbrain is located. Smoking, the prevent or delayed Parkinson's disease, but obviously they have a 16 times higher chance to get lung cancer. We don't recommend smoking to prevent Parkinson's disease. Now, redrive those cells into a particular target.
Welcome back to BroncoTales Season 2. This episode, we are hosting Dr. Kim with the Neuroscience program. Welcome in. Thank you for being here.
Thank you for inviting me.
Yeah. Would you like to just introduce yourself to the audience really quick?
Hello, my name is Hwan Kim. I'm a program director of Neuroscience program, and we opened our bachelor's BS program of Neuroscience last summer, and this is a brand new program, and we are quickly expanding and growing. So we're recruiting actively numerous faculty from biology, psychology, kinesiology, and others. And now biomedical engineering and local hospitals, including St. Luke and St. Al and VA Hospital, they are part of the neuroscience program.
Yeah. Awesome. And you primarily, in your lab, research Parkinson's disease, correct?
Yes.
What is the main cause of Parkinson's disease?
So Parkinson's disease, first of all, maybe 10% or around 10% of patients are genetic, means familial Parkinson's disease, meaning that they were born with some genetic mutation. So some of them is considered autosomal dominant mutation, then you will definitely have Parkinson's disease in your 40s or early 50s. Some cases, autosomal recessive, meaning both alleles, one from mother, one from father, both of them are mutated, then you will have Parkinson's disease. Obviously, much rarer than autosomal dominant cases, but then unfortunately, the onset is earlier, so patients are having Parkinson's disease in their 30s instead of 50s. But then vast majority of PD cases are sporadic. They don't know why they got Parkinson's disease. So then usually the patient's onset is around 60 or mid-60s. So later.
On average it's
60s.
60s? Okay. And then early onset would be 30s?
Right, 35-ish.
35,
but then it is rare. Only 1 or 2% of Parkinson's patients are early onset.
And what are some of the first, kind of symptoms that people start to see, whether it's regular onset or early onset?
They start to complain about stiffness. Oh, I used to, walk around comfortably, but now my body is holding stiff. Even my back is kind of crouching a little bit. And then some of them start to show mild tremor. So then, something's not right. But then some of them may not take it seriously because, hey, I'm 70 years old and, my body's stiff. I guess I'm getting old. But that's not real reason now. In real reason behind is they're losing dopaminergic neurons in the substantia nigra, where the midbrain is located. So then dopamine synthesis and release would be reduced. Therefore, their motor control is deteriorated already. But then too late.
And how do you see kind of what you just described in research, but also like a diagnosis? Like how does, what scans, kind of what diagnosis do the people have to go through? So
the best way to do it is functional MRI. So we know where dopaminergic neuronal the location is and where dopamine release is happening. So based on functional MRI, certain area like substantia nigra or pudicodamine, where dopamine release is normally happening, that area would be quiet, less active compared to healthy control, age-matched and gender-matched. So then the neurologist can comfortably conclude, you'll have Parkinson's. You are having or progressing Parkinson's disease. So then we can test kind of opposite way, giving some medicine whether that actually helped their mobility or not. So they can verify that, yes, you are having Parkinson's disease.
And what are some of the ways, if any, that have been discovered to help prevent or reduce, maybe push back how, like the age range that you can kind of
We don't
start developing
have
symptoms?
an excellent way to prevent Parkinson's disease. At this point, that's why numerous scientists are studying, Parkinson's disease. So many reports suggest that nicotine, like smoking, the prevent or delayed Parkinson's disease. And based on statistics, yes, smokers, have lower chance to get Parkinson's disease, but obviously they have a 16 times higher chance to get lung cancer or other disorders. So, we don't recommend smoking to prevent Parkinson's disease. And after having Parkinson's disease, mild exercise, including yoga and others, would be beneficial in terms of reducing stiffness.
What about in terms of like the tremor, like, hand-eye coordination exercises, kind of things of that nature to kind of strengthen those?
Probably helps. And this, we can say it doesn't hurt because, the mild exercise often stimulate dopamine release. So, and these patients feel better with that. And we don't have any curing method for Parkinson's disease, and no real, the curing the medication either. So everything available in the market for Parkinson's disease patient is symptom alleviating medicines. Nothing to do with, curing the disease.
Right, just to help kind of ease the symptoms.
Yeah. Right.
So what, we kind of talked about the causes. What does the Kim lab do in terms of Parkinson's disease?
So we try to understand pathological mechanisms of Parkinson's disease, and ideally we want to identify, the novel therapeutic target. So particularly I'm interested in cellular senescence. So we are getting old, and cells are getting old, and some point cells stop proliferating, then we call senescent cell. So in neuroscience, we didn't pay attention to cellular senescence much because neurons do not proliferate. But 90% of cells in brain, they are glial cells. They are locally proliferating. So that's why now we pay attention to cellular senescence aspect of Parkinson's and Alzheimer's disease pathology. And we found that brains are experiencing a pathological condition or progression is already happening, then more senescent glial cells were detected based on our the observation from human samples and even mouse model. We published one paper related to that, and currently we are running a couple of projects related to that.
And what, how do you gather this research? What are kind of the steps do you kind of have to go through, and then you're able to look at these things?
So we have to get or establish our the research tools, which is typically mouse models or cell lines or cell models or human samples. So sometimes we get human Parkinson patient samples, especially substantia nigra, the frozen samples, and we analyze whether our target or biomarker would be higher or lower from the patient compared to age-matched control. So then that's one way to validate, okay, this is a reasonable target or not. And we manipulate animals, like typically mouse models, for that purpose. So we can genetically manipulate that. Something toxic, then we can overdrive genetically, or we can inject alpha-synuclein preformed fibrils into the brain where the dopamine release is happening. So then 2-3 months later, those animals are experiencing Parkinson-related symptoms. And we can see progression is kind of quickly happening after some point. So we use those animal models for our research tools. So then, for example, cellular senescence, we want to remove senescent cells. So then we orally gavage senolytic, which is inducing apoptosis for senescent cells to remove toxicated cells to prevent neurons. So we are using cell model or sometimes a stem cell model or primary cell culture, which means isolating brain tissue from baby brain, and then we culture in vitro. Or, as I said, we're using mouse model, transgenic or injection model.
So you're kind of artificially stimulating the brain?
Right. Kind of we generate the neurotoxicity in the animal brain to mimic Parkinson's condition.
Okay.
Then we can apply potential therapeutic application, whether that actually reduce the toxicity or prevent the neurons or not. So we can test that in mice.
And what's the overall main goal of your lab?
Well, the ultimate goal is based on our findings. As I said, potential therapeutic target. We want to use the target for therapeutic application. So we are also working with a biotech company and kind of discuss, and we found some novel target, and they design either small molecules or other like antisense RNA. Things like that, then we apply to the pathological mouse model, and we test whether it is convincing or not really. So then we kind of maneuver, not working, then we redesign potential drug and possibly a completely different target. So we are mainly interested in developing therapeutic application for Parkinson's.
So not solving, how you get Parkinson's and that, but mainly helping patients that already have the disease.
Right. So it can be two different, the route. One way is if we can preventive medicine would be the best.
Right.
But again, we don't have a good pre-diagnostic marker for Parkinson's disease, so we have a very limited, the access. Second thing, after patients are showing symptoms, we have to help them in terms of alleviating symptoms and possibly halt pathological progression. That's the goal. If we can halt, then possibly we can reverse pathology a little bit. Then everything will be in the right direction because they probably feel better, they can move better, and possibly preventing secondary damage, which is like dementia or others, depressions and dementia. Because 25% of PD patients in the later stage, they're experiencing depression and dementia.
So that kind of just builds off of or develops from Parkinson's.
Right. So if we can kind of halt the disease progression, then probably the downstream other pathology would be prevented as well. As I mentioned, like a dementia or depression.
Which would almost be a form of a cure of some sort.
And this...
It wouldn't stop it, but you're trying to get enough to make it easier to live with.
Right. So, and this we prevent the further damage. Maybe not reversing from the pathology they developed, but at least halt right there, and then prevent further damage, then their quality of living would be much better.
Right.
So that could be very important application as well. But ideally, we can, prevent from the beginning. Then we need to identify, the pre-diagnostic tool, which is also lacking in Parkinson's and Alzheimer's.
And what drew you to kind of choose this research and field?
Well, initially I studied molecular biology for my bachelor's degree, and then I studied biotechnology for my master's degree. But then right before finishing my master's degree, my mom got stroke. And I realized I studied biology or physiology for several years already, but I didn't know much about brain. And that was kind of turning point. I'm interested in neuroscience. I want to study neuroscience. So I decided to study neuroscience for my PhD So that was my initial engagement to neuroscience. But then what I studied for my PhD thesis was sex differences in brain. Why males are the responding differently or behave differently than females. What kind of gene expression would be different? That was my thesis topic. But then my last, the project before I left UCLA was Parkinson's disease. Patients are a little bit more prevalent in males than females. So then whether any genes on X chromosome prevent, the Parkinson's disease onset from female side. So we studied that genetically using mouse model, and then I, was deeply engaged to Parkinson's disease. So then I got my first postdoc job at Johns Hopkins for studying that, and then I moved back to California to study Parkinson's disease continuously at the Buck Institute. So since then I studied Parkinson's disease for the past 20 years or a little bit longer. Yeah.
And was that first at UCLA, was that your choice to choose Parkinson's, or was that?
That was actually kind of mutual agreement between myself and my PhD mentor. I liked that project, so that's why I was willing to take over the project. So yeah, it's a mutual decision.
You also kind of have mentioned in your lab that you used or research deep brain stimulation. What does that look like in for a human brain, and how can that help with Parkinson's?
I haven't studied deep brain stimulation in my lab yet, but I'm interested in doing that. But I have interacted with several DBS, the transplanted patients, Parkinson's patients, and their response was very positive. And I have interacted with the neurosurgeons and neuroscientists in several institutions, including Rutgers and University of Florida and Hopkins. So it is very promising. So simple idea is surgically implant small microchip into, in our case, into the substantia nigra, because we know where our target is, which is substantia nigra, where dopaminergic neurons are dying. So that surgically implant there, and then putting the pulse chaser or regulator underneath of the skin with a battery-operated system. So then when patients feel stiff, then they can push the patch, then electrically stimulating the microchip in with the brain, then they feel better. And again, based on my interactions with the patients, they say they like it, and their quality of living is much better. But still, it's an expensive procedure because the major invasive surgery is required, and insurance companies probably not support that. So sometimes it costs like $500,000, things like that. But a lot of positive responses by patient. But one caveat is we don't know how it works really. Simply just stimulating whole region, even though it is pretty narrow, but how it works really, we don't know exactly. We assume yes, stimulating whole neuron, then the dopamine release would be stimulated as well, but is it really the mechanism? Probably so, but we don't know the exact mechanism. Second thing is, it doesn't really prevent the further disease progression. So it's not really curing the disease. So that's another downside.
Like the quality of life.
Right.
And it doesn't sound like, do you know how many people, or it's not a very common?
Not that common yet, because again, it's not
super expensive.
Right. It's not covered by insurance company, and also it's not really disease modifying, application, although it's substantially alleviating their symptoms, but it's not really curing the disease. So very limited access and limited availability for average patients.
So would you say it's probably not the best way to kind of route to move toward? Or do you think in the future it would be?
I think a lot of patients are highly recommend.
Okay.
Because many of them are saying that their quality of living is substantially better after surgery. So I think it is probably a good way to go. But again, it's not really disease, curing application. So, theoretically better way is a stem cell research. Simply patients lost dopaminergic neuron 50%, whatever or more. Then the iPSC, so induce pluripotent stem cell from your own skin, and then in vitro differentiation into dopaminergic neurons, and then they can get that neuron in vitro differentiated, in this case, dopaminergic neuron into their own substantia nigra, and then let them reconnect. So very limited application of stem cell because like same thing, extremely expensive, and insurance doesn't cover, and not many the labs out there can do it consistently because culturing stem cell could be very finicky because cells, the live, thing, sometimes they, differentiate it slightly differently, then outcome would be different too. So a lot of variables in this application, but a friend of mine at the McLean Hospital, which is a Harvard Medical School, he reported in New England Journal of Medicine about 30 years ago with a success story. So I think a lot of promise for this application, but we need to decide how we can lower the cost. One way to do it is genetic, not necessarily transplanting my own cell, just like a custom-made cell line, but they are dopaminergic neuron, and then transplant, and then after surgery we need to get immunosuppressant, to take the cell, right? Someone else's, the neurons.
So you're artificially growing, or what you're describing is like an artificial growth of stem cells outside of the body.
Yeah, in vitro. On the dish we can culture either my own cell or someone else's cell, and then differentiate them into in this case, dopaminergic neuron, and then transplant to the target area, and then wait. And one the caveat of this approach is whole protein aggregation is the main cause for Parkinson's, Alzheimer's, and other neurodegeneration. So then pathological condition is not cleared yet, but then giving new neurons, some people believe those new neurons will take up protein aggregates, quickly within several months or within a year. Then they probably lose those new neurons again. Then is it worth it to spend $500,000 or whatever?
So it might not be
So we still have several hurdles to overcome, but theoretically this is, the most, the promising approach because this the application can cure the Parkinson's disease and Alzheimer's.
And what do you mean by differentiate the cell into the dopamine receptor? So
the iPSC means the induced pluripotent stem cell. So we're using our own skin cell because skin is one of the few cells still proliferating actively, even though patients can be in their 70s. But then we have to de-differentiate and then redrive those cells into a particular target, like dopaminergic neuron. So we need to get dopaminergic neurons, right? So that's why we convert skin cell to dopaminergic neurons.
So it's
that's what I mean
transforming.
That's what cell. That's what I mean by differentiating into a particular target. Let them lose skin cell, characteristics, and then becoming more stem cell characteristics. Then we can drive them into dopaminergic neuron. It takes 2-3 months on dish, but we already have protocols like that. Not me per se, but the in our community this is well established. So that part is relatively easy, but the surgical the transplant takes time and costly, and also after surgery nothing's guaranteed. So it's still it's a risky surgery.
I know you probably get this question a lot, and you've kind of mentioned but if you were to get Parkinson's disease, run through the process that you would go through, or would you do it any differently than most?
Practically not a lot of options.
Yeah.
So because of, the older medicines available now is symptom alleviators. So generally the L-DOPA, which is a precursor of dopamine, that works very well for most PD patients.
That's a medication?
That's medication.
Okay.
It has been established for a long time, more than 3-4 decades. But it does have a side effect. So L-DOPA, 90% of patients are saying, oh, L-DOPA works, yes. But taking L-DOPA for 9 years, then 90% of the patients are developing L-DOPA-induced dyskinesia, kind of out of control. So then they need to get another medicine to suppress this kind of side effect. That's why nowadays doctors are not prescribing L-DOPA or L-DOPA-related medicine like Cinemat, because we all know it does have a side effect. So then, starting with a mild medicine like a dopamine agonist, like stimulating receptors, not really, touching on dopamine directly, or inhibitor, which is blocking dopamine's normal degradation, then dopamine level would be slightly higher in the system, then the patient feel a little bit better, but it's not excellent medicine. It's very limited, efficacy for many patients. So then what is the choice I would have if I'm a patient? Again, not much, but mild exercise based on several study, yoga or other mild exercise help dopamine release. So probably that I will do that, and then if possible delay L-DOPA taking, because we know it does cause the side effect. So again, then very limited choices.
So that's kind of not a last resort, but one you don't want to immediately go to.
L-DOPA probably not, and also Parkinson's disease patients, they live for a while. Many patients live more than 10 years, 15 years. So you have to think about long-term effect. So as I mentioned, L-DOPA works, but you have to think about what about 5 years, 10 years later. So that's why many neurologists are very reluctantly prescribing L-DOPA-related medicine. But then patients are complaining, I still can't move around well. Can you help me? Then okay, I'll give you low dose of L-DOPA-related medicine. But then, unfortunately, you have to increase the dose over time because of the tolerance issue. So same amount of or same dose of medicine doesn't work after a couple of years. So then you have to increase the dose. So then elevating the chance of having side effect, LID or dyskinesia. So that's the dilemma. Patients like it, initially, but then they will soon experience side effects. So then what is the kind of
balance?
Right, balance between them. So that's why many patients are actually kind of refusing taking any medicine for a while. But after some point, they have to take it. Otherwise, it's too difficult to move around daily basis. So depending on the patient, in general, their health level is high. Other than low level of dopamine, then maybe they can delay a little bit for taking any major medicines. But some of them is already tumbling down, so then likely they may not even live another 10 years. Then probably better to take a medicine. So if I'm a patient, then I would consider that. How many years I have realistically? I have probably 10 years or longer. Then I will reluctantly taking any major medicine. If I don't feel like I have that much time, then I'll take Cinemat or L-DOPA-related medicine.
Well, I don't have anything else.
Okay, good.
Yeah, thank you for joining me, and thank you everyone for watching. It was very interesting to learn kind of the behind the scenes of the neuroscience and your lab.
Thank you for having me, Sam.
Yeah, thank you.
All right, cool. Thank you.